Biology 101, Lecture 10. Hello, students. Today we're going to talk about meiosis. Meiosis, yay! This is a very special type of cell division. It's not like mitosis, but very similar. But there are some changes, and the idea is that we need to reproduce ourselves. And so when we try to reproduce ourselves, there are two ways to go about doing this. One is asexual, where the single individual passes all of its genes to its offspring without the fusion of any gametes. And so, in truth, it's just cloning its own self. So a clone is a group of genetically identical individuals from the same parent. That would be asexual reproduction. But in sexual reproduction, two parents give rise to offspring that have unique combinations of genes inherited from them. And so what we are going to look at are the actual things that do this work. That would be the chromosomes. The chromosomes carry all the genetic material. They are, when they're about to divide in cell division, that's when they become visible, and that is known as the karyotype, which is an ordered display of the pairs of chromosomes in a cell. The two chromosomes in each pair are called homologous chromosomes or homologs. Chromosomes in a homologous pair are the same length and shape, and I'm going to show you an exception, and carry genes controlling the same inherited characteristics. So, but when we look at the human karyotype and the human genotype, like right here, we see chromosomes 1, 2, 3, 4 lined up, and they look very, very similar, same size, about the same shape, except when you go all the way to the bottom line and you see chromosomes X and Y, not the same size at all. The Y chromosome is awfully small, so there's somewhat of a mismatch there. Those are the two chromosomes. They're called the sex chromosomes. They determine the gender of the individual. They're also called X and Y chromosomes. Human females have a homologous pair of X chromosomes, so they have two X chromosomes. But human males have one X and one Y chromosome. The remaining 22 pairs of chromosomes are just called autosomes because they have nothing to do with the gender. Each pair of homologous chromosomes includes one chromosome from each parent. So there are 46 chromosomes in a human somatic cell. 23 pairs, that means two sets of 23, one from the mom and one from the dad. A diploid cell, which we can always write in parentheses as 2N, has two sets of chromosomes. One N from one parent and one N from the other parent. For humans, the diploid number is therefore 46. So 2N is equal to 46, which means N is just 23. Haploid. Now, what is considered haploid number of chromosomes? Well, the gametes, which could be either the sperm or the egg, they contain a single set of chromosomes, and they are called haploid. They just have the one N. For humans, the haploid number is 23. Each set of 23 consists of 22 autosomes and one single sex chromosome. So in an unfertilized egg, the sex chromosome would be the X chromosome, but in the sperm cell, the sex chromosome could be either. It could be either Y or X. So we're going to look at those. And so there are two different types of sperms that are produced. One carrying an X, one carrying a Y. But the eggs are all the same. They all have X's. So in this picture, what we're looking at is some mythical individual with only 3 pairs of chromosomes. Okay, so his 2N number or diploid number is 6, and the red chromosomes are, we're going to color them and say that they're from the mom, and the blue ones are from the dad. Okay, so here what we're seeing is these chromosomes have already begun dividing. So although the red chromosome, the very long red chromosome, and the similar looking very long blue chromosome, they're one pair. They're homologs. They have already started dividing into two. So this is the beginning of division. So not the best picture, but we're just looking at it. And then there are two medium-sized chromosomes, and then there are two itty-bitty tiny little chromosomes. They're just showing that cell division has begun, and we're going to look at what happens to the chromatids in the homologous pairs. We are going to talk about both. We've already talked about mitosis, but we're going to contrast it with meiosis, because meiosis is awfully similar to mitosis. The only difference is there is a reduction of chromosomes. If we didn't reduce the number of chromosomes, pretty soon, in one generation, you'd have double chromosomes. The next generation would be even more than that. Then you can see where this is heading. So a lot of the chromosomes would, in like 3 generations or 4 generations, you would have these mutated people, which or offspring, which are not in the original design, and they were not supposed to be like that. So at meiosis, the chromosome number is halved. One, to preserve the integrity of the chromosome number and the species, and 2, to provide variation and diversity. So we're going to look at both ideas a little bit carefully. Fertilization is the union of the two gametes. The sperm and the egg will unite, and they become one. So the sperm will donate its chromosome component to the egg, and the egg will no longer be an egg, but it will be called a zygote. So the fertilized egg is called a zygote, and it has one set of chromosomes from each parent. The zygote produces somatic cells by mitosis and develops into an adult. So once there is a zygote, it will just do cell division or mitosis and become an adult. But at sexual maturity, that same adult will have, depending on its gender, ovaries or testes, and those are going to produce haploid gametes, not the rest of the body. Gametes are the only types of human cells produced by meiosis rather than mitosis. Meiosis results in one set of chromosomes in each gamete, so it's half the number. Fertilization and meiosis alternate in sexual life cycles to maintain chromosome number. Sometimes these life cycles are pretty distinct. Gametes are the only haploid cells in animals. They're produced by meiosis and undergo no further cell division before fertilization. Gametes fuse to form a diploid zygote that divides by mitosis to develop into a multicellular organism. And here is a schematic showing you that. So every time you see 2N, that means it's diploid. Every time you see N, that means it's haploid. And you see a regular old mouse, or it's a multicellular diploid organism. He spends his life in the 2N stage. However, his genetic organs, gender organs will undergo meiosis to produce one N, or one set of chromosomes, which will then join up with somebody else and at fertilization, and then produce a zygote which has the same 2N number, or double the number of chromosomes as compared to the gametes, and that cycle will be complete. Plants and some algae do exhibit an alternation of generations. So this is kind of cool. In vertebrates and mammals, you don't see this. They're all the adults are diploid. But some plants and algae will actually alternate generations. One's haploid, the next one's diploid. So the life cycle includes both a diploid and a haploid multicellular stage. The diploid organism, called the sporophyte, makes haploid spores by meiosis. Each spore grows by mitosis into a haploid organism called a gametophyte. So the gametophyte makes haploid gametes by mitosis. And then ultimately you'll have fertilization of these gametes, which will result in a diploid sporophyte. And you get the cycle to be complete, which is complicated, but not so much. If you look at the schematic, what you see is at the very top, let's start with the haploid multicellular organism, or the gametophyte. And so it already has half, so it's not going to undergo meiosis anymore, but it will undergo mitosis and become a nice little plant. However, when it needs to fertilize, it will fertilize with another gametophyte to produce a zygote, which is diploid, and that stage is called the sporophyte, because that's the only stage that will actually produce new cells, new organisms. The gametophyte cannot produce more of itself. It can only produce, get to be a bigger size, but it can't make more gametophytes, like more children. Whereas the a diploid organism will produce more of itself. So that's just in some plants and maybe some algae, but not in the rest of the kingdoms. In most fungi and some protists, the only diploid stage is the single-cell zygote. They have no multicellular diploid stage. So they just exist in haploid. The zygote produces haploid cells by meiosis right away, and each haploid cell grows by mitosis into a haploid multicellular organism, and the haploid adult produces gametes by mitosis. So you never get a multicellular diploid stage. And here is a picture of that. So at the top of the picture, there's the haploid unicellular or multicellular organism, which is represented by N, and grows by mitosis and then fertilizes, produces a diploid zygote, which immediately halves its number of chromosomes by meiosis, and then it exists mostly in the haploid state. So that's most fungi and some protists. That's what they do. Depending on the type of life cycle, either haploid or diploid cells can divide by mitosis. However, only diploid cells can undergo meiosis. So that's one big difference. Although haploid cells can divide by mitosis, the only cells that can undergo reduction of chromosomes would be the diploid cells. The halving and doubling of chromosomes contributes to genetic variation in offspring. Like mitosis, meiosis is preceded by the replication of chromosomes, which would be in interphase. Meiosis actually takes place in two consecutive cell divisions. The first cell division is called meiosis 1, and then we have meiosis 2. And after the two cell divisions, you end up with 4 daughter cells rather than 2 daughter cells in mitosis. But each daughter cell in meiosis has only half as many chromosomes as the parent cell. Chromosomes duplicate during interphase, and the resulting sister chromatids are closely associated along their lengths, just like they do in mitosis. This is called sister chromatid cohesion. The chromatids are sorted into 4 haploid daughter cells ultimately. And here we are. Here is a picture showing you interphase. At the top, you have one pair of chromosomes. So this individual has only 1 pair of chromosomes. It's hypothetical, but that's how it is. So the blue is coming from the dad, and the red is coming from the mom, and it's one set, one homologous pair. Well, during interphase, when it's going to start to divide, just before that, the chromosomes are going to start duplicating, and they become sister chromatids lined up but attached with a centromere. So it is still a diploid cell, still considered a diploid cell, because the chromosomes are not separated. Under the microscope, they just look kind of fat. So at this point, meiosis will start. And at this time, the homologous chromosomes are actually going to separate. They are not going to go to each pole, as in mitosis. They're going to separate without the centromere splitting. So one duplicated chromosome with the sister chromatids attached by the centromere is going to go to one cell, and the other one is going to go to the other cell. So they are now haploid. So they have half the number of chromosomes as the parent cell, which had 2. So now each cell has 1 chromosome. That's meiosis 1. That's what happens in meiosis 1. But in meiosis 2, cell division occurs again. But at this time, the centromere will split, and those chromatids will actually migrate to different poles of the cell, and you end up with 4 daughter cells, each of them having half the component of the original parent. So each will have 1 chromosome. In meiosis 1, also you had the 1 chromosome, but actually it was duplicated, and the number, although technically the number is halved, but there's still a duplication going on, and the centromere is still attaching to duplicated homologs. So although you can say that the number has already been reduced, and it has, we still need to split that centromere and go on to meiosis 2 when it is actually complete. So to sum it up, here we have the whole cycle. In meiosis 1, what happens? The homologous chromosomes separate. If you start on the left-hand side, what you see are sister chromatids and homologous chromosomes. They're just there. The nuclear membrane is disappearing. And you see metaphase where they're lining up at the center, at the equator. And then in anaphase, they're starting to move away to the 2 poles. Well, here, the 2 poles are going to only get a sister chromatid, a homolog, with the 2 chromatids attached still by the centromere. And you get cytokinesis, you get 2 cells, and then right after that, immediately after that, we are going to have meiosis 2, where prophase starts, and then again metaphase, where things are lined up at the center, and anaphase, where things are separating, and telophase, and cytokinesis 2. I do want to point something out. If you look very carefully, in anaphase 1 of meiosis 1, you notice a small change in the chromosomes. So although at metaphase they look the same, exactly the same, what happened at metaphase was that some genetic material got swapped over to the other side. And you see little parts of chromosomes becoming blue and red because they swapped over parts. So although they're homologs, they're not really homologs at all. They're completely different now from the original components they had. They've mixed. So when you look at the anaphase 2 of meiosis 2, you notice that very clearly. It's very distinct and clear that genetic material has been swapped from one chromosome to another. So we'll go through the steps one by one. Meiosis 1 is called the reductional division because it reduces the number of chromosomes per cell. Division in meiosis 1 occurs in 4 phases: prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis. So in prophase 1, we have the same organism with 3 chromosomes in his genetic material. Each homologous pair undergoes synapsis and crossing over between non-sister chromatids with the subsequent appearance of chiasmata. So you see that a little bit more clearly in metaphase 2, although you do see some beginning of that in prophase 1. in metaphase 1, the chromosomes line up as homologous pairs on the metaphase plate, and then in anaphase 1, the homologous chromosomes separate from each other. The sister chromatids remain joined at the centromere, and you can actually see parts of the chromosomes that have been swapped because they're in color. In prophase 1 of meiosis 1, what happens? After interphase, the sister chromatids are held together by proteins called cohesins. In early prophase 1, each chromosome pairs with its homolog and crossing over occurs. X-shaped regions called chiasmata are sites of crossover. The non-sister chromatids are broken at precisely corresponding positions, which is very neat, because otherwise there would be a mismatch. A zipper-like structure called the synaptonemal complex holds the homologs together tightly, and DNA breaks are repaired very fast, joining DNA from one non-sister chromatid to the corresponding segment of another. So there's actually a complex which will hold the chromatids together while they're crossing over. And the DNA is breaking, but it's repaired very rapidly. Here's a picture of what actually is happening. If you look at the left-hand side, let's start from there. Picture number 1. So first, the DNA is going to break. The cohesins are there. The centromere is there. And then there are DNA breaks again. What you see is 2 strands of DNA, and they're parental sister chromatids, and they're a pair of homologous chromosomes. However, when you look at picture number 2, which is at the bottom of it, you see this synaptonemal complex forming, which is got all these little green dots and all this complex stuff where the breaks are. And then all of a sudden in picture number 3, what you see is this zipper-like structure that is holding everything together while these breaks are occurring. And the chromosomes are actually swapping over. They're going from one side to another, and you see that very clearly in the blow-up part with the arrow. And in picture number 4, what you see is it's done, it's completed, and you have the chromatids, the sister chromatids, but now they have different stuff on them. They didn't have that originally. So we're going to look at it one more time. Look at the DNA breaks. We're going to focus on those, and then we're looking at the complex forming where all these little green dot-like things are there. And then in this picture we have the zipper-like structure helping keep everything together, holding everything together while there's a crossover, and the DNA breaks are repaired, and you end up with chiasmata, and you form new chromosomes. In metaphase 1, pairs of homologs line up at the metaphase plate with one chromosome facing each pole. Microtubules from one pole are attached to the kinetochore of one chromosome of each tetrad, and microtubules from the other pole are attached to the kinetochore of the other chromosome. In anaphase 1, pairs of homologous chromosomes start to separate. One chromosome of each pair moves towards opposite poles, guided by the spindle apparatus. Sister chromatids remain attached at the centromere and move as one unit towards the pole. In the beginning of telophase 1, each half of the cell has a haploid set of chromosomes. Each chromosome still consists of 2 sister chromatids. Cytokinesis usually occurs simultaneously, forming 2 haploid daughter cells. In animal cells, a cleavage furrow forms. In plant cells, no cleavage furrow, but a cell plate forms, and we looked at that in mitosis. No chromosome replication occurs between the end of meiosis 1 and the beginning of meiosis 2 because the chromosomes are already replicated. Here we are, meiosis 1, looking at it again. So now if we look really carefully at the very first picture on the left-hand side, you see chiasmata forming. Yeah, and then in metaphase 1, what you see is the lining up of the chromosomes in the equator, in anaphase 1, and they're separating, they're already different. They're separating in telophase 1, they have ended up in their destinations. Meiosis 2 has the same phases, same 4 phases. Division in meiosis 2 also occurs in 4 phases: prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis. It's very similar to mitosis. In prophase 2, a spindle apparatus forms. In late prophase 2, chromosomes, each still composed of 2 chromatids, move towards the metaphase plate. In metaphase 2 of meiosis 2, the sister chromatids are arranged at the metaphase plate. Because of crossing over in meiosis 1, the 2 sister chromatids of each chromosome are no longer genetically identical. The kinetochores of sister chromatids attach to microtubules extending from opposite poles. In anaphase 2 of meiosis 2, the sister chromatids separate. The sister chromatids of each chromosome now move as 2 newly individual chromosomes towards opposite poles. In telophase 2 of meiosis 2, the chromosomes arrive at the opposite poles, nuclei form, and the chromosomes begin decondensing. And then cytokinesis separates the cytoplasm. At the end of meiosis, there are 4 daughter cells, each with a haploid cell set of unreplicated chromosomes. Each daughter cell is genetically distinct from the others and from the parent cell. And so here we are with mitosis 2, where the sister chromatids are being separated out. The crossing over has already occurred, and we're now just going to separate them out into gametes or haploid cells. So you see the picture. It is exactly what we talked about before. Mitosis conserves the number of chromosome sets, producing cells that are genetically identical to the parent cell. But meiosis reduces the number of chromosome sets from 2, which is diploid, to 1, which is haploid, producing cells that differ genetically from each other and from the parent cell. So here's a comparison of mitosis and meiosis side by side. If you look at the left-hand side, that's mitosis. You have the simple prophase, metaphase, anaphase, and telophase. And what you see is in prophase, the chromosomes are duplicating. Again, the diploid number is 6. So it has 3 pairs of chromosomes. And you see all the chromosomes are duplicated and lined up in metaphase at the equator. But in meiosis, right away in prophase 1, we're seeing a difference, okay? So there's already crossing over. There's chiasmata forming, and there's crossing over genetic material in homologous pairs. So in metaphase 1, yes, they are going to line up. But notice, first of all, that they are different already. And also notice that they are not lined up like they were lined up in metaphase 2, metaphase of mitosis. Over there you had 1, 2, 3, 4, 5, 6 chromosomes and that were already divided, lined up. Here in meiosis 1 you only have 3 sets of chromosomes lined up, and so the homologs are going to separate at anaphase, and they hop on over to the other side of the cell, and then you have sister chromatids that will divide again, and in meiosis 2 you end up with, at the end, with 4 different daughter cells, each one distinct and each one different from its parent. So when you're comparing mitosis and meiosis, please start with prophase. Please notice that the prophase is where the changes are, and also metaphase is pretty different, because if you look at the equator in mitosis, it is completely different from the equator in meiosis, although it's the same animal with chromosome number of 6. And here is a comparison of mitosis and meiosis in anaphase and telophase. So in anaphase, in mitosis, what you see is chromosomes moving away to opposite ends. But in meiosis, the homologs are separating, and that is where you end up with 4 cells instead of 2. Okay, here is a summary. It is in words what we just discussed and looked at. In mitosis, the DNA replication occurs in interphase before mitosis begins. And in meiosis, it also occurs during interphase before meiosis 1 begins. In mitosis, there's only one division, including prophase, metaphase, and anaphase and telophase. But in meiosis, there are 2 divisions of the cells. In mitosis, there is no synapsis of homologous chromosomes, but in meiosis there is, because the crossing over has to occur. Chiasmata have to be formed to hold the sister chromatids together. In mitosis, the number of daughter cells and genetic composition, let's talk about it. Well, it's going to be 2 daughter cells with genetically identical to the parent, same number of chromosomes. After meiosis is complete, you have 4 daughter cells, each with half the genetic component and each distinct from not only just each other but from the parent. And what is the role of mitosis in the animal or organism's body? Well, it will enable it to produce more cells for growth, repair, and so on, to make it bigger. And in some species, it is also responsible for asexual reproduction. And it also produces gametes in the gametophyte plant. Whereas in meiosis, what is its role? It produces gametes. That's it. Or spores, if it's a sporophyte plant. It reduces the number of chromosome sets by half, and it introduces genetic variability among the gametes or spores. Another compare and contrast. The property in mitosis of DNA replication. Well, it occurs in interphase before mitosis begins, and there's only one division. There's no synapses of homologous chromosomes, and you end up with 2 cloned cells of the parent, which allows you, the multicellular animal or plant, to arise from a single cell and to be able to grow, repair, and so on. However, in meiosis, DNA replication, although that occurs in interphase also, the number of divisions are 2. And there is a crossing over, and there is a synapsis of homologous chromosomes, and you end up with 4 haploid, genetically different cells. And the role of meiosis is to produce gametes. That's it. Just to produce gametes for genetic variability. The unique events in meiosis 1, there are 3 of them, and all of them are listed right here. So the first one is the synapses and crossing over in prophase 1. Homologous chromosomes physically connect and exchange genetic information. Number 2, homologous pairs at the metaphase plate. That is unique. And number 3, separation of homologs during anaphase 1. These are all unique events in meiosis 1. Mutations, which are changes in an organism's DNA, are the original source of genetic diversity. Mutations create different versions of genes called alleles. Reshuffling of alleles during sexual reproduction produces genetic variation. 3 mechanisms contribute to genetic variation. 1 is the independent assortment of chromosomes. 2 is crossing over, which we just looked at. And 3 is random fertilization. Homologous pairs of chromosomes orient randomly at metaphase 1 of meiosis. In independent assortment, each pair of chromosomes sorts maternal and paternal homologs into daughter cells independently of other pairs. The number of combinations possible when chromosomes assort independently into gametes is 2 raised to n, where n is the haploid number. For humans, the haploid number is 23. So that means 2 raised to 23, which means that there are more than 8 million possible combinations of chromosomes in just humans. So here is possibility 1. 2 equally probable arrangements of chromosomes of metaphase 1. Notice how they're arranged. They're identical, right? So you could have, remember, the blue is for the dad and the red is for the mom. Well, at metaphase 2, notice what is going on the right-hand side. It's different. And so the daughter cells are different. Crossing over produces recombinant chromosomes, which combine DNA inherited from each parent. Crossing over contributes to genetic variation by combining DNA from 2 parents into a single chromosome. So you get the best of both worlds. In humans, an average of 1 to 3 crossover events occurs per chromosome. So that's a lot of variety. That's why children are almost never like their parents. Here is a schematic of that with an actual electron micrograph showing you prophase 1 of meiosis, where crossing over is actually occurring, and you can see the chiasma. So we're not just making it up, it actually occurs, and we've caught it red-handed. Random fertilization adds to genetic variation because any sperm can fuse with any ovum. The fusion of 2 gametes, remember each of them have about 8 million possible chromosome combinations from independent assortment. Well, they will produce a zygote with about 70 trillion diploid combinations. Okay, so therefore, there's a tremendous variation resulting from sexual reproduction. And then, on top of that, crossing over adds even more variation. So each zygote is truly unique. It has a unique genetic identity. Natural selection results in the accumulation of genetic variations favored by the environment, and sexual reproduction contributes to the genetic variation in a population, which originates from mutations. So organisms can increase their genetic diversity through horizontal gene transfer.